Yo, what's up? It's Coach Hyde coming to you live from the Digital Learning Day. Hope y'all are all awake by now. Y'all know y'all ain't awake. Come on, wasting life. Get up. Get the day started. Anyway, all right, so what this is, just trying to do a real quick rundown of the macromolecules. Going to leave some things out, but the goal is to kind of fly through this, make you the shortest video I can to help you get ready for the test coming up after our fall break. So 4 macromolecules we're focusing on: carbohydrates, lipids, proteins, nucleic acids. Not a whole lot of depth on nucleic acids. All right, our main focus is on the carbs, the lipids, and the proteins. So we're going to kind of run through those as best we can. There's going to be 4 big polymers that we're going to talk about for carbs, 4 polymers for lipids, all right, and proteins. I guess we focus on enzymes the most, but I mean they could do all kind of stuff, right? So the enzymes and then the folding process are the main points of the proteins. Nucleic acids, the song. I don't know where the paper of the song is, right? Check out the song. There's a video of it. It's called the DNA song, right? Made a sugar, phosphate, and a base. Anyway, so starting out, carbohydrates. So this kind of section up here is in carbohydrates. Right? Carbs are sugars. Another word we use for sugars are saccharides. Right? And then all these kind of end in ose. So glucose, fructose, sucrose, maltose. All those are sugars. They end in ose. All those are simple sugars. Right? Then, so those will all be our monosaccharides or our disaccharides. Every time the announcement comes on. But anyway, so these are our oses, our glucose, our galactose, our fructose we talked about. One carbon, our one carbon chain, carbon structure here, carbohydrate, right, made out of carbon and water, or 2, di meaning 2. So if we were to continue to build these sugars and make them more and more and more complex, add these monomers together, then you'll get to our polysaccharides. And there are 4 polysaccharides. Now, let me get my little pointer stick. 4 polysaccharides, right? Chitin, cellulose, glycogen, and starch. So chitin, that is used for structure, cell walls in fungi, and then the exoskeleton in the phylum Arthropoda. So that's the structure, carbohydrates focus there. Cellulose, also a structure carbohydrate. These are cell walls in your other plants. So cellulose and chitin are both used for cell walls, maybe plants and fungi. And then chitin is also the exoskeleton. Glycogen. So this is for our quick energy stored in our muscles and our liver. This is what we burn before we start burning fat, right? This is what we use readily available energy in our bodies, and animals too. Starch, right? This is energy storage in plants. Kind of same purpose as glycogen in humans. There's 2 kind of strands we need to know. So we got our basic long single chain, don't branch off. That is amylose. And then you have the one that's kind of more complicated, branching off in different directions, amyloseptin. So this is the shorter word, amylose, right? For the more simpler, or simpler word for the more simpler structure. This is a bigger word, kind of more complex, right? More complex word for the more complex carbohydrate, right? For the starch. So that's it. That's our carbs right there. Done. Okay. Then we'll get down a little bit. Next up, I don't know how focused this thing is. Lipids, right? Lipids are fats, right? Got a lot of lipids right here. Okay. I'm storing energy for the zombie apocalypse. It's going to happen. I'll be ready. I'm not going to starve to death like them skinny folks. Anyway, lipids are fats, right? There are 4 polymers. There are more complex fats that we'll focus on: wax, triglycerides, cholesterol, and phospholipids. So we're not really going a whole lot of depth into wax. Just understand wax is lipid, candle wax, earwax, right? Beeswax, wax paper, then wax on leaves, seeds, kind of protect it, water repellent type stuff. But it is a lipid. Lipids are insoluble in water. We also have cholesterol. Won't go into that, but it's a big 5 carbon ring structure of lipids. But they are the precursor for steroids, and some hormones are steroids like testosterone, estrogen. What's the stress one? Cytosol? Yeah, that one too. Something like that. So then we kind of go into these triglycerides and these phospholipids. These are our triglycerides right here. Our triglyceride is made up of a triglyceride made of glycerol, right? You got glycerol in there, right? So we got glycerol in there. The tri is standing for our 3 fatty acids. These little squiggly lines represent hydrocarbons, a little different than carbohydrates. So carbohydrates is carbon with water, right? Hydrogen and oxygen. Hydrocarbons, so these, right? These are just carbon and hydrogen. That's the only thing on there. And they might have a little acid in. They do, but whatever. So these fatty acids can either be saturated or unsaturated. The big key difference here, and you can't really see it without forward, but this one, right, has that double bond there. All right, so that double bond kind of kinks it. It makes it where it's not full of hydrogens, whereas this one are full of hydrogen, right? Every carb has at least 2 hydrogens on it. The end carbs have 3 or whatever. So these carbons only have 1 hydrogen each. So it's undersaturated with hydrogens. Our saturated fats, think of them as like long, straight chains. They're easy to pack together. So they are solid. These are solid fats like butter, animal fats, stuff like that. Those from animals. Our unsaturated fats are healthier, right? They're easier for the body to break down. They kind of get into that double bond, break it apart, and make energy. These are liquids. These can be found in plants and fish. Our other thing over here on that side, these are phospholipids. So when you hear phospholipids, think cell membrane. All right, here's our structure. It's the same basic structure as a triglyceride, except we would lose one of these fatty acids, and we put a phosphate group there instead. So you end up basically with this fatty side and this phosphorus side. And why this is unique is because the phosphate side is polar. It's polar, it's charged, right? So it interacts with water and other polar things, whereas this part down here is not polar. It is no charge, right? It does not interact with polar things, but it interacts with unpolar things. So when they start to come together, they hook up like this, right, and form this bilayer, this double layer of phospholipids. This is what our cell membrane looks like. So you end up with this kind of charged area here, charged. So this, and then the inside is hydrophobic, right? Which means it does not interact with water. So it creates this layer that kind of repels stuff and it prevents things from going through. But it also can interact on the outside and inside with polar things, which is great for communication. But yeah, cool. I'll tell everybody you're out here, but you can't come in. You know what I mean? So that's it for lipids. Next, trying to go fast. All right, I wanted to get this done in 10 minutes. It ain't looking good. So proteins. Proteins are made of amino acids. So our monomer for proteins is amino acids. These are things that end in INE, right? Amino acids end in INE. So like leucine, tyrosine, stuff like that. We put them together, right? We make proteins. Proteins is our polymer. Now there's a process from getting from amino acids to protein. And what it is our nucleic acids holds the code to how to put together the amino acids. We'll put the amino acids together. When we bond the amino acids together, that's called a peptide bond. All right. Once you start making these peptide bonds, these amino acids, you have a peptide, and as you continue to grow, eventually it becomes a polypeptide. So our primary structure is the formation of this polypeptide, right? Amino acid, amino acid, amino acid, bonded together by a peptide bond. Then our secondary structure, the polypeptide becomes these alpha helix, right? Helix is like circular spiral staircase situation, and the beta plated sheets. It's kind of like a crumpled accordion situation, right? Then we go to our tertiary structure, which is these alpha helixes and beta plated sheets on this big polypeptide. Now we start to get the interactions of the R groups, and maybe they repel, maybe they stick together, maybe they bond, sulfur bridges. I mean, there's all kind of stuff that happens there. And we end up with this globular or fibrous, globular like globe-like, fibrous is like strands, proteins. And then we can combine these tertiary structures and we'll get our quaternary structure, And then we have our protein. We have our polymer, which is the protein. So examples of proteins, enzymes, of course. So things that end in A's. Catalase is a great one. The one we talked about, we did a lab about enzymes, right? One way we describe enzymes is with the lock and key method. So if you think about a lock, you think about a key. A key only works with one lock. I know there's exceptions, but let's just simplify it and say a key works with one lock. It's not a master key, just a normal, simple man's key, right? Works with one lock. Now, you can put that key in the lock, unlock it, lock it, pull it out, essentially as many times as you want, right? Over and over and over and over again. You never use up that key, you never use up that lock. So an enzyme is similar. What an enzyme will do, just like lock and key, right? It will bond substrate. So the substrate floating around in this area situation, right? And eventually, most likely, the substrate, when conditions are right, will find this hookup partner and it will bond, right? And you'll end up with a new compound. But what an enzyme does is it like initiates the reaction, right? And Lorenzo was old that the enzyme told the body or helped the body metabolize the extra saturated fat. So what it's doing is it's grabbing it, saying, "Hey, we got to break this stuff down, man. We got to break it apart." Or what an enzyme can do is it can go grab something and say, "Hey, you need to be with this one. Let me hook y'all up," right? And then it makes this, right? Then it lets go. Okay, it goes and finds another one over here. "Hey, I got one. Let me take you down here, hook you up with your partner." Right, got it. So enzyme is not used up in the reaction, right? It's it's facilitating the reaction, it's initiating the reaction, it's making the reaction easier to occur, it's speeding it up. It's a biological catalyst. Hope that makes sense. So other examples of proteins here: egg whites, albumin, egg whites. I mean, pure protein, I guess. I don't know, it might have some other little stuff in there, but it's like 17 calories, pure protein, good stuff. Your eyes, color and stuff, protein. Now, your DNA codes the proteins, right? But the proteins, your DNA codes for these things to be the way they are, but the protein is, is what it is. Your muscles, some motor proteins that carry stuff around. Remember the little vesicle thing? Hauls the vesicles around up the cytoskeleton bridge and all that. There's carriers, milk, cesium, I don't remember, something in milk, right? For baby calves to grow. Blood cells, hemoglobin, bonds the oxygen's protein. All right, so pretty much everything is protein. You need protein for pretty much everything. All right, nucleic acids. DNA, deoxyribonucleic acid. RNA, ribonucleic acid. These are our nucleic acids. This is our genetic material of our bodies. They are made of nucleotides. Right? A nucleotide is kind of this situation over here. It's made of a sugar, phosphate, and a base. And your base are the things you've heard about, like adenine, cytosine, guanine, all that. Okay. Last kind of two things running through: dehydration synthesis. Right. So you see A plus B equals C. So you're taking two things, you're joining them together, and in the process, water is removed. Right. So you see water out. The kind of opposite of that, hydrolysis, is where you take something and you break it apart by inserting water, essentially, right? You need water to go in there, and this water reactions and stuff is going to break it apart, right? Lysis means to burst, I think, burst or split apart, cut in half, burst, I think, as far as the root goes. If cells are lysing, that is bad. It's not good. Hydro meaning, so we're using water to bust it. So, again, we left out a couple things. It's a 14-minute video, so it's pretty long, but I mean, it's like 2-3 works of material right there, or weeks of material. So, hope this helps y'all get ready for the test. Hope you're enjoying your digital learning day. Hope you're learning on your digital learning day, not fishing and hunting and stuff. That sounds fun too. All right, see y'all tomorrow.